Thanks, Hong, and thank you all for coming today and for the opportunity to speak. What I was hoping to do with my talk was to do a brief introduction to IMES and to the Health Sciences and Semiconductor program, and then I'm going to talk a little bit about some of the work that we're doing in our own research group that connects back to the overall theme. As you'll see, I'm much more genomics and molecular biology oriented, but it will couple into the front end of the Health Sciences Semiconductor program. So as Hong just mentioned, I wear many hats and I'm affiliated with many institutions. The one I wanted to start off by representing today is the Institute for Medical Engineering and Science, or IMES. IMES is really an institute at MIT that's focused on bringing to bear our expertise in science and engineering on problems in human health. So we sit at the intersection of these areas and act as a bridge to surrounding industries, hospitals, and partners to make sure that we can develop technologies that are going to improve human health and wellness. And so our broad remit within MIT is to really be an integrative force for all human health research at MIT. And so if you're looking for something in the space, you can always feel free to reach out to me. I'm delighted to connect you. This is not only true for academics, it's also true for hospitals and industry. We like to be a strategic portal where we can. And the other thing is that we are home for an educational program that's split between Harvard and MIT that trains engineers in clinical medicine and doctors in engineering rigor. That has incredible students that come out of it that you should recruit, or if you're looking for interns, we could talk about it. So I'm going to talk about a couple things today, and in my own work, I'm going to focus on cancer. But I want to be clear that across IMES, we have about 270 faculty members between Harvard Medical School and MIT that span a wide variety of different organ systems, disease processes, technologies, and healthcare concepts. Many of the biggest names at MIT, are within our bounds. And so if there's things that you're looking for, from drug delivery to, organoids, we can talk about any of that. Just find me offline. And as I said, we do have this incredible educational program where we train MDs from Harvard and PhDs from MIT. And these are the students that aren't content to just get a degree from Harvard or one from MIT. They need to have a degree from both. So if you're looking for fantastic students, I'm happy to connect you to some of them. So as an institute at MIT, our big goal is to focus on scientific areas that bring together all of our faculty, into shared foci. We have 6 that we primarily focus on. One is medical imaging, one is pediatric innovation, one is AI for health, one is health sciences and semiconductors. We also have climate and medicine, and the wild card is space medicine. I'm happy to talk about any of these, but for today, I want to focus on 2 very briefly before I get into my own work. And given the topic of today, I'm going to start with health sciences and semiconductors. So the real vision for this program that's co-led by Ahmad, Tomas, and myself is to shape the future of personalized healthcare through innovation. Given the incredible strengths in semiconductor technology in Taiwan, I don't have to introduce to you that there's a revolution happening in personalized healthcare. Advances in semiconductors have made it possible for us to measure tremendous amounts of information from different biological systems and to think about real-time monitoring and closed-loop intervention. I think that with all the excitement here, we're just in the exponential growth phase, and I would posit that MIT is poised to help lead this revolution, given the strengths that we have in semiconductors and edge AI, the clinical and translational centers that we have, including the ability to do clinical research in IMES, our state-of-art facilities at MIT Nano, and the incredible local ecosystem within Boston that connects us to multiple different hospitals and additional institutes. So with this in mind, our broader goal within the Health Sciences Semiconductor program is to really bring together health science, artificial intelligence, and semiconductor technology to build minimally invasive measurement techniques to address human health care and wellness. Our big vision is to do this as a full stack approach, where we not only think about one part of it, but we go all the way from sensors to data collection to making the most out of the data that we can through edge AI as well as more conventional approaches, and turn these into closed-loop solutions where we actually deliver things back to the patients through actuators. Now, this is not something that we think that we can do alone, but in our ecosystem, we think we're very well poised to have impact by partnering with local biotechs, labs, and really the patient communities that we hope to serve. And our overall goal is not just to do this in Boston, but to think about the needs of people everywhere and to partner with people all around the world, and it's why I'm so excited to be here today. A lot of what we've been doing within this program is to start to build community and to start to get projects moving forward. So symposiums, seeds funding, partnerships with industry. I'll say that we've had a number of meetings thus far at MIT, at the Broad. We have an upcoming one at MGB. If you're interested, I'm delighted to tell you more about it, but it's really bringing together people across domains to talk about real urgent problems. And we're working on places that will come up in some of the talks today, from minimally invasive ways of measuring hemodynamics and hemoglobin to new technologies in polymers that can help with nanopores, to things that Tim might touch upon in terms of monitoring of inflammatory biomarkers. And that ties a lot into the things that I'll talk about. And we're really keen to have conversations with you all to think about where we might partner at all the different stages, from early device development to actual implementation. So if you have questions, please just find me at break or Ahmad, or check out our website. I do want to say really quickly before I dive into my own science that we have coupled with this an entire AI for health program. I think I don't have to tell everybody here that AI and machine learning have tremendous promise for human health, but there are major barriers to implementation where we need to solve problems that are critical to building trust in the systems, from improving interpretability and trustworthiness, to figuring out how to make sure that we understand failure modes, to figuring out how we can leverage the data to have robust solutions that you would trust to give to your loved ones or to yourself. And so in parallel to the Health Sciences and Semiconductor program, we've been standing up a large community of individuals across the Boston area between the Broad, MIT, IMS, and many other places, focused really on having real impact. And within IMS, our big goal is always to do innovative science, but the ultimate goal is always impact. And so what we've been really focusing on are places where these technologies can be brought to bear to have real changes to the way in which people are getting care in the clinic. And we're focusing on key areas of cardiovascular disease, women's health, cancer, and infectious disease. So if you have questions, please ask me about that. All right, and now for something entirely different. The way that I got involved in this program originally was that, I do come from a nanobiotechnology background, but over the past several years, I've spent a lot of time developing and applying technologies to really understand the basis of disease. And now that we know how to measure things, the big question in my mind is, how do we monitor them and how do we intervene? And that's why I'm so excited about this program here. So as a brief introduction to the work that we do within my own research lab, what I'm really interested in is what defines health. All of us want to be healthy and to have balanced tissues. But if you were to ask me, what does that mean at a cellular and molecular level? I wouldn't be able to tell you. But we know it's important because if we have imbalances, like too much immune activity, we can get tissue damage and death, say an inflammation. And if we have too little immune activity, we can become susceptible to opportunistic infections and tumors. So what unifies the work in my group is a desire to understand what constitutes balance. What are the deviations that are induced by exposures to things in the environment, such as pathogens or pollutants, and to think about how we can restore health as a therapeutic or buffer it in the first place as a prophylactic. Now, the reason it's been such a good time to think about tissues and balance and all these things is that over the past 10 to 15 years, there have been transformative advances in what we can measure in cells. This has brought together advanced molecular biology and micro and nanofabrication to the point where we can now extract incredible amounts of information. So it's no longer one protein or one RNA, but we can actually measure tens of thousands of variables per cell. We can measure the transcriptome, so everything a cell is thinking. We can look at the DNA of an individual cell. We can measure what regions of that DNA are accessible. We can look at proteins. We can do these together. We can innovate additional technologies. And I'm happy to talk about that. But the most important thing to me out of this is sometimes just because you can measure something doesn't mean that you should. And so a fundamental question we've been trying to ask is, what should we measure, why, when, and what does it do for us? And so I wanted to talk about this in the context of cancer. So, if we think about cancer, we always think about it as a disease of mutations. And so our fundamental precision medicine approaches are based upon this idea of identifying mutations that exist. And so if you had a tumor, if I had a tumor, or a loved one had a tumor, I would rush into the lab and I would try and sequence it to figure out what mutations are there. I would then try and build a model, an organoid, a cell line, a mouse, and screen a bunch of drugs to figure out which one would be useful for that individual so that I could get the best possible outcome. Now, you might ask, if this is truly precision medicine, how well has it worked? Well, it's worked okay. About 40% of patients have actionable alterations, and there's some really beautiful examples here. If you think about like the BCR-ABL fusion, the Philadelphia chromosome, and ponatinib, but the problem is that most patients don't have actionable mutations, and in many places, people evolve resistance to these targeted therapies. So the question we've been asking ourselves in cancer is, what is the control's response and resistance in addition to DNA? So the funny thing is, I'm not trained as a biologist, even though I teach it now, so I always try and think about the problem as an engineer. And what you realize when you do this is that we're not trying to kill a mutation in cancer. What we're actually trying to do is kill a cancer cell. And so that changes what we have to go after in our overall approach. So the funny thing is that when you think about DNA, there's a long way from DNA to the cell. So each of us has about 37 trillion cells in our body. They all contain, to a first approximation, exactly the same DNA. Yet that DNA gives rise to everything from the endothelial cells that line our blood vessels, to the immune cells that patrol our bodies, to the neurons that you're using right now to listen to this conversation. And how does it give rise to that diversity? Well, it's through epigenetics, so accessibility in the DNA, interactions with the environment, and reading this DNA, this set of instructions, as a dynamic RNA expression state. And so if this is the diversity that you can get under normal contexts, then perhaps not surprisingly, in the context of cancer, the same mutations can give rise to multiple different cancer cell states, some that are immune responsive, some that are drug sensitive, and some that are resistant to therapy. And so what we became interested in is understanding this coupling between DNA and state and thinking about how it related to treatment. So to tackle this, we took an approach which is very indicative of the work that I do and that IMS does, where instead of trying to do it individually, we partnered with people from all across Boston, bringing to bear innovative technologies based upon semiconductors for measuring mass and growth rate, to single-cell genomics, to functional assays, and coupled that with advanced models and primary patient samples and computational strategies. And so I'm just going to give you one little vignette, and then I'm going to use it to explain how we're thinking about this more broadly. So the cancer that I want to focus on is a devastating one. It's pancreatic ductal adenocarcinoma. This is a really bad cancer, but really interesting from the genetics vantage point because it has very uniform mutation profiles. So about 90% of people have KRAS mutations. You probably know that there's a drug against one such mutation, G12C, which works for 1% of the 90% and was a big deal. And that Revolution Medicines has recently announced a new drug that goes against pan-RAS mutations. Unfortunately, that extends life by about a factor of 2, but it doesn't constitute a cure. So what people have done is they've looked at this cancer through other lenses. And what they've discovered is that the cancer can take two major expression states. It can express genes that look stem-like, which we'll call basal, or more epithelial-like, which we'll call classical. And the reason people care about these signatures is because they have prognostic value. Neither is good, but basal is worse. And so as I think about things like health sciences and semiconductors, here are signatures, things that we'd want to measure as a way of understanding prognosis. So we have genetics, we have states. How do all of these couple together? Well, in looking back at what we already had, we went and took all our primary samples, we took our cell lines and our organoids, and what we discovered was that the mutations that we had in patients were well modeled in our labs. But when we looked at these states, states that these cells can occupy, it turned out that the diversity that we saw on the left didn't show up in our cell lines or organoids. Our cell lines were always basal, and our organoids were always classical. And what this taught us was that our models don't do a good job of recreating states, but also that there's something about the way in which we make our models that influences these programs. So in trying to figure out what was going on, we thought about many of the things that you're probably thinking about now. Maybe it's selection, so the way you grow something dictates what you get. Maybe it's plasticity, so they can change under different conditions. Maybe there's lots of interactions when you go to 2D versus 3D. So to try and really benchmark this and understand what was happening, we built a simple pipeline with our partners, Dana-Farber. And this may seem funny, but it's a really important experiment where we take tumor biopsies, we dissociate them and characterize single cells using deep technologies that we have access to, and in parallel, make models and characterize those to understand what is gained and lost. And so what we ended up doing was a large study, and I'm just going to run you through the highlights of it and then extend it out. But it was driven by Pete, Andrew, Hannah, and Tree, and previously published. So the basic gist of what we did in this experiment was to start off by taking primary biopsies and characterizing them using these technologies. So here I'm showing you a map of high-dimensional gene expression space. We're measuring 20,000 plus variables for every single cell, so trillions of data points overall. And each cell is a point, and we group cells based upon similarity. We've also used spatial technologies to figure out where the cells are and to do high-content digital pathology. The long story short out of all of this is that actually these tumors can be basal, classical, or you can have a state that expresses both. So this tripartite graph that we've seen in many different cancers, suggesting that maybe there is some plasticity. We also discovered that there are distinct tumor microenvironments that surround each of those cancer cell types. We can now go play the same game and figure out what's going on with our organoids. And we apply the same technology, and what we discover is that there's a strong bias against the basal program in our models. In thinking about how we make models, what we realized is that the way we make models is we take these tumors, which are in this very stressed environment with limited nutrients, where there's constant immune attack, and we put them into a dish and we pamper them with lots of different reagents that make them grow and make them healthy and happy. And what that does is, not surprisingly, it changes their phenotype. So what we discovered was that if we simply started playing with how we cultured these cells so that they looked like they were in environments more similar to a tumor, we could recreate all of the states that we saw normally in patients. Similarly, if we took molecules from the surrounding microenvironment and added them, we could change where the cancer cells were in a predictable way. Now, rather than this just being an experiment where we wanted to play God, the real thing that we were trying to do here is figure out whether or not this was important. And the reason I'm telling you about all of this is because it is. So it turns out if you have the same genetics, but you put your cancer cells in the basal state, they're 100 times less sensitive to chemotherapy than when they're in the epithelial-like classical state. Now, it's not just that the stem cancer cells are less sensitive to drug. It turns out that there are different sensitivities in different states. So the epithelial-like state is sensitive to chemotherapy. The stem cell-like state is sensitive to MAP kinase inhibition. And what's so nice is that we can now select drugs based upon biomarkers. And it turns out that this will also tell us what people are going to respond to when we go back and look at clinical trials. So the individuals that have classical tumors are the ones that respond on the right, whereas the ones with basal are the ones where you have outgrowth. So this is great. And I like to think about it as sort of a vertical stack in PDAC. We start off with an understanding of what the patient's tumor looks like. We build models, and then we go and try and find vulnerabilities. But what we'd like to do is we'd like to build something that's applicable to multiple different cancers. And so how can we do this? Well, we can go do the same thing and start building reference maps for multiple different cancer types. And working with the National Cancer Institute in the United States, we went through and we characterized a lot of their samples to figure out what was going on. The really interesting thing that comes out of this is even though these are entirely different cancers, many of them express that same basal program and therefore probably are sensitive to the same MAP kinase inhibition. The next thing that we want to know is how can we create models, thinking about the role of intrinsic genetic factors versus these environmental factors in dictating states. So for intrinsic factors, what we've discovered are ways of screening to figure out which transcription factors and internal cellular components drive particular programs. So we found that by overexpressing specific programs, we can turn on the basal state or the classical state. And we've developed some nice screens as a way of figuring out when genetics will dictate this, working with Feng Zhang and some of his students. And I'll highlight that along with this, we found markers for each individual state, not just RNA, but proteins, so that we can either pull those cells out, monitor them, or target them with antibody-drug conjugates. Now, on the extrinsic side, it's a little bit harder, because when you deliver a plasmid to overexpress a gene or a program, you can look at a barcode and you can figure out what you put in. When you put chemicals in solution, it's a little bit harder. And so to develop a solution here, we innovated a new pipeline where we figured out how to mix drugs together and then figure out the impact of each one individually. And the intuition is that we can build a linear model to figure out effects as long as we only assume that a couple of things interact. And this lets us take those same pancreatic cancer organoids that I was telling you about before, screen the impact of every single signaling molecule that might be relevant, discover new ones like IL-4 and 13, things associated with allergy that we never would have looked at there, and then go validate them to prove that when we do them individually that they do what we want. In screening things, what we realized here was that there was a lot that we were missing, because if we look at IL-4 and 13, I never would have imagined thinking about allergy and pancreatic cancer. And the reason for that is that when we think about it, we turn on with IL-4 and 13 about 133 genes in PDAC, of which between 5 and 0 overlap with the signatures that we have in the literature. But getting back to that map that I gave you in the beginning of one DNA sequence giving rise to multiple cancer cell states or multiple cell types, you can imagine now that in a cancer cell that has different epigenetics, different proteins, different interactions, you could get a very different response than in a T cell in the nose during allergy. It turns out the signatures that we have are prognostic, whereas the other ones aren't. Getting back once again to things that we might want to monitor. But the other thing that comes out of this is that it's not just IL-4 and 13. The other signatures are also, limited overlap. It's just that there's enough there that we can discover them when we do testing. So what this made us realize is that in thinking about what we would want to monitor, like in the way in which Tim is doing stuff with CRP and IL-6, that we really don't understand what the majority of communication signals within our body do. We know that our cells have to communicate to accomplish their goals, but we don't know what signals are important. And that's because the majority of our literature really focuses on a small set of cytokines, and we really only focus on drugging a small set of them. The way in which we have selected these things is not how relevant they are to disease, how relevant they are to expression. We've looked at them most based upon when we discovered them. So with the oldest cytokines being the most studied, where we're building on the literature we already know. And even from the small set of molecules that we know, there's incredible therapeutic opportunity because there's billions of dollars every single year in sales targeting these particular molecules, either overexpressing them or blocking them. And from the vantage point of what we're talking about here, one of the reasons I really like these molecules is that they're extracellular targets, so very easy to target with an antibody, very easy to measure in solution, and we can use well-established protein technologies to go after them. So in thinking about this, what we realized was that we needed to engineer a new technology to think about what signals do among cells. And so I love to cook, so I've loved being here because I like going around and eating. And one of my students said, what we really need is we need like the same thing we would do at a dinner party where you make a lot of little dishes and everybody gets to taste. So we need tapas. And so Constantine went and built a protocol where we could basically create lots of different proteins by arbitrarily producing them very rapidly in a dish from cell-free lysates with high quality among humans. And so it's one of those things where previously you would have thought you could not make cytokines outside of cells, but now we can. We get high quality and yield across multiple different proteins. We can produce hundreds of different things across different classes, from the cytokines and interleukins that I talked about before, to engineered variants, to nanobodies, to whatever you would like. And all of them are functional. So our IL-2 works just like the commercial variant. Our interferon gamma drives the same gene expression responses, and our TNF-SF10 drives the same apoptosis, or cancer cell killing. So this is great, but we didn't start making these hundreds of proteins just to figure out what we already knew. We did it so we could screen. And what we discovered when we applied this to human blood, thinking about biomarkers, are that there's incredibly interesting patterns that emerge from all of this data. So here I'm showing you about 3 million cells where we've screened about 300 proteins across 4 donors. I don't have time to talk about all of it, so I'll just give you a couple really quick highlights. There's some really nice patterns that come out with different classes of proteins that are similar to one another being coupled, things that make sense like IL-4 and its engineered variants being together. But there's also interesting things that we discover about cell type specificity for each of these responses, so that if we want to get something out of one cell type but not another, we can. The data makes sense. Our IL-6 induces an IL-6 signature. Our IL-2 induces MYC targets in IL-2. Our interferon alpha induces alpha signature and then gamma, and our gamma does gamma and then alpha. But it's not that we're just discovering things that we already knew. We're actually discovering new targetable biology. So here's a protein that has basically only 50% homology with IL-2, but it drives incredibly strong cytotoxic responses among NK cells. You can see it in the TNF and the interferon gamma in the granzyme B, but it does not drive regulatory responses, so no FOXP3. So this looks like an incredibly good immunotherapy that we could begin using. And what's so interesting about it is it doesn't do this in all cells. It's NK cell specific. So it doesn't activate this in CD8s or in CD4s. So that just gives you the tip of the iceberg about the kinds of molecules that we could start using for therapies. But the other thing that we can do, really thinking about signatures and monitoring through the health science and semiconductor lens, is we can take all these incredible atlases of disease that we've built, measure what's going on in them, characterize things out of patients like I did in pancreatic cancer, go and deconvolve what might be driving that based upon our signatures, and use this as a way of predicting what's relevant to block or what we should be monitoring on a device. And so what we end up having are these beautiful maps where we get lots of different signatures, lots of different cytokines. We can discover that there's relationships that make sense to us. So here's a bunch of cytokines that drive these really strong cytotoxic responses like IL-2 and Super-2 or Neo-215. But we can also discover molecules that are really important, for example, that drive strong memory responses that you might want in a vaccine, whether it's a cancer vaccine or an infectious disease one, which we can now deploy therapeutically or we can use for monitoring of efficacy. So diving back into the end of this to wrap up, I think you have an idea now of how we're screening things. In the same way we were compressing cytokines, we can compress drugs. And we've screened hundreds of drugs simultaneously on the same sample, and we can pull out what each one does. We can overlay on top of this interesting things like what do these environmental factors do? How do the genetics work? And how does the intersection give you, response so we can figure out what are the right interventions for each person? And you might ask, why am I so excited about this approach? Why do I think it's so valuable? It's because when we start looking at the data, what we discover is that the state-specific targeting is probably going to transform precision medicines. So if you look at our cancer cell lines here on the bottom, you can see I've got some breast cancer, some bladder, some head and neck. It doesn't matter what the tumor of origin is. If I look up top at the RNA expression state, it tells me in the middle what genes it's going to be sensitive to. So if I do CRISPR knockouts of specific genes, I will get the same response across all tumors that have a basal program and all ones that have a mesenchymal program. And so fundamentally, what I'm telling you is that cancer cell state is really a measurement of what pathways a cell has active at a given moment. And so by targeting those instead of mutations, we can do a better job of effectively killing the cancer. The important thing becomes really understanding these cancer ecosystems. So I think about this as completing the square or building a foundation model, if you would like, where we want to understand what drives pan-cancer ecosystems so that we can think about how to engineer models, but also how when we add drugs, we can limit cancer cell plasticity to drive more effective outcomes. Out of all of this, we're getting incredible databases and signatures that are really helping us to figure out what to advance to the patient, what we might want to deliver. So as we bring in new signatures of what diseases look like in our perturbation libraries, we can feed these into deep learning approaches that will enable us to discover lead compounds that we want to follow up on, but also to go test those to close the loop so that we can get better signatures and get to better therapies quickly. So in talking today, I've really focused on the idea of cancer, but our group works incredibly broadly. And so we do infectious disease, we do inflammation, and we're applying exactly the same strategies to each of these. So thinking about inflammatory bowel disease, it's very similar in the way in which one might go about it. So with that, I just want to thank our current team. They're incredible. They do science. I just get to fly to fun places and eat incredible food. But I'm happy to have any opportunity for them to tell you about what they do. And I want to thank our collaborators. This is a small subset. At last count, we have collaborators in 35 countries on 6 continents. And I primarily want to thank our patients and families, because a lot of what we do in IMS and in the lab is centered on delivering impact to patients. And we recognize how stressful it is for them to work with us, particularly when they're going through their own medical issues. And so a lot of our goals are to help deliver care to them and to feedback what we've learned so that they can benefit from it. Thank you very much.